All right, welcome everybody to this webinar on CRISPR therapies for sickle cell and Bio-Rad's Out of the Blue CRISPR kit. I'll be your guide in this webinar today. My name is Damon Tighe. I'm Bio-Rad's curriculum and training specialist for the Western region of the United States. But before we get any further into this, let's just kind of do some housekeeping things. For those of you that have not attempted a webinar on Zoom before, everyone's mics is kind of zipped up, so that way I'm the only one talking. But as you have questions that roll in, there is a Q&A function that you can put your questions in. And I have a handful of Bio-Rad folks here helping me this evening. So Aaron Callis, Aaron Hansfeld, and Ian are all three in the background helping with questions as they come in. And they'll try to answer what we can during the talk. And then if there's ones that I need to attend to at the end, we'll kind of take some time to then go over those and go into some more detail. There's also a chat. You can put stuff in there, but it's way easier if you put it in the Q&A because you can kind of check off whether or not the question has been handled and things like that. So please, if you can put them in the Q&A, it's a little bit easier to handle than trying to dig them out of a very long and rolling chat, especially when there is a couple hundred people in here at once. So my name is Damon Tighe. Somebody's asking for subtitles. I'm trying to open those up right now. Zoom is not allowing me to add the captions in for some reason. If you are somebody that needs the captions, we will record this and I can send you a recording later with the captions built in. Zoom's preventing me from being able to do that right now in the transcript. So sorry about that. I thought that usually that's been automated in the past, but that's not allowing that tonight. My name is Damon Tighe. I'm Bio-Rad's curriculum and training specialist for the Western region of the United States. And if you're not familiar with Bio-Rad, Bio-Rad first is a biotechnology company, but they have a really small educational division that works at taking what's going on basically in kind of contemporary biology and then putting it in formats that's easy to bring into your classroom. And they're in a really kind of cool niche place to do this because Bio-Rad's making these technologies and these tools for researchers. And so we can literally do what's going on in the major company and then break that off into ways that makes it easy for you to bring that into a classroom and do them hands-on, using real stuff, real tools, real reagents, real instruments. We have a small group within that educational unit that goes out and does hands-on training and then helps basically assist on curriculum kind of fits and development so that we can tailor these things so that they fit into each individual classroom. And that's our curriculum and training specialists. So I'm one of those. I cover the Western region of the United States, basically kind of Colorado to the coast. Lee Brown covers the Southeast, Tamika Stubbs, the Northeast, and then Aaron kind of covers all of these places in between that don't get enough touch points from other people. But together, they're just part of the team that's here to help you bring kind of modern biology into your classroom in digestible ways. So for this evening's webinar, what we're going to cover is a lot of content, but I'm going to try to get it to you at the levels I think is right for who's here. So we're going to cover a little bit about the FDA history, mainly just so that we can talk about the clinical trial process, because this is going to be key to understanding how do these tools that are developing out of the kind of the CRISPR landscape actually become therapies that change people's lives. To see the history kind of tells you why the FDA is designed the way it is and where it's flexible and where it can continue to change in the future in order to adapt to changing therapies, technologies, and drugs. We'll then jump into some of these CRISPR therapies. Actually, we'll look specifically at one of them because I think that it illuminates a lot of what's going on in the landscape. And this one is specifically going to make solutions for sickle cell and beta thalassemia patients. We'll dig into the weeds nice and deep there. And then we'll kind of pull back and then we'll look at something that you can do actually in your classroom, hands-on with your students to demonstrate the power of CRISPR in your classroom. And the beauty about that is it's something you can do with very, very little instrumentation. And it gives students the real feel of what it is to do CRISPR and why it's such an exciting new technology. And then with the time we have left, we'll do a Q&A. Where I want to start though is with this photograph here. So on the right there, you see Victoria Gray, and she's actually packing her bags down in Forest, Mississippi, where she's from, in order to go up to Nashville, Tennessee, where there's a clinical trial taking place. Victoria is actually the first person to enter a clinical trial that's going to use CRISPR to solve sickle cell disease. And Victoria is a really interesting person in a lot of ways. As somebody that's in a clinical trial, you could easily just go out into the background. Nobody needs to know who you are. But Victoria's decided to be very kind of like vocal and public about her process, which is amazing because it gives us all insights into what it's like to be a patient, the power that these technologies have as far as even just the hope it is for patients of solving these diseases. And so we're going to kind of follow her pathway a little bit tonight in order to understand what's going on with these therapies. So Victoria had her first sickle cell crisis at 3 months old. So her grandmother was actually giving her a bath and she was just crying like she was in pain. And then the crying wouldn't stop. And the grandmother realized that these were cries of pain. This wasn't just normal, like, I'm just fussy. So they ended up taking her to the hospital. And at that hospital visit, 3 months old, they realized that Victoria had sickle cell disease. And this is something that then would plague her the rest of her life, these kind of sporadic pain events to the point where there's hospitalization involved, and a really rough regimen of ways to then deal with it, including like prescribed narcotics like fentanyl and oxycodone in order just to do pain management. Victoria puts a lot of these into sound bites, which is kind of nice for us to kind of conceptualize what it is to be a patient. So she says, I had aching pains, sharp pains, burning pains, you name it. That's all I've known my entire life. I was hurting everywhere my blood flowed. And it was so bad that Victoria just couldn't really lead a normal life. She always had the dream of becoming a nurse, but in her kind of academic progress of becoming a nurse, she ended up getting hospitalized so frequently due to pain that she wasn't able to kind of complete her studies. And at some point, she found out about this clinical trial that was going to move forward, basically offering a potential solution to sickle cell disease for the first time. And she jumped at it. And partly for herself, right, to be able to live a normal life, but even more so that it's, as she says, it's not just for me, but for other people, this would be mind-blowing. Oh my God, I can't imagine the lives that could be saved if this thing actually works. And so this very brave person is basically going to allow us to kind of see this process from the patient side, which I think is something that we don't see a lot with many kind of clinical trials. And so this is something, the story that will evolve throughout the next, I'd say 5 to 10 years, that's going to be worth kind of keeping an eye out for. But in order to understand how we got to a point where Victoria can even enter this clinical trial, we need to understand a little bit about the clinical trial process. How does the FDA bring safe and effective drugs to market that can revolutionize a patient's condition? And there's lots of intricacies into this process. We're going to do like a medium level dive into here because you could spend a semester in here with all the idiosyncrasies that goes on in this process. But high level, there's a look at the manufacturing of the drug or the therapy that the FDA is going to look at. And then they're also going to look at basically the scientific evidence that you have something that is safe and effective. But how did we even get to this place in history where this is the process? Well, a little bit of this comes back to where the United States and the world used to be before these sort of things were instituted, which is what I would call generally the unregulated era, which is if you had a condition and you said, boy, I've got this thing, there were plenty of people in the marketplace that said, hey, I've got something I could sell you that would solve that, some sort of snake oil. In fact, that is still in our vernacular today, this idea of snake oils. But the snake oils, to be honest, weren't the really scary things. The really scary things were some of these things that were other drugs that were used for their stuff. They're being kind of repackaged and then sold in order to solve other problems. These led to a lot of death and a lot of dangerous places. So one of the most famous ones is Miss Winslow's soothing syrup. It's just a nice concoction to get your baby to go to sleep and stop crying. And it just happens to be just morphine and alcohol. So there were plenty of children that never woke up from their soothing syrup back in those days. And it really wasn't until the start of the last kind of 100 years that the FDA really kind of gets its birth. And for the most part, it is reactions of the government to things going on in the marketplace that create a unsafe environment for citizens or for people in the country, or at least in the United States. So this really kind of gets kicked off, the FDA, I would say, gets kicked off around 1906. This is the whole meat packaging industry. people adding borax to meat in order to make it, stay together better, even though it's really, really toxic. You have Upton Sinclair, the jungle, and ultimately this gets you the Pure Food and Drug Act that establishes the FDA in 1906. There's hundreds of instances that give you all the nuance of the FDA, but we won't get into all of them. I'm just gonna highlight 3 of them. The second one that gives us the FD&C Act, the Food, Drug, and Cosmetic Act, that basically says this stuff that enters the market has to be safe, comes out of this reaction to the sulfanilamide poisoning of 1938. We have this drug that's already being used, but they wanted to get it in a liquid form that was easier to give to children because you could sweeten it and things like that. You could mix it with like raspberry syrup. And they gave it to a chemist, and the chemist said, oh, I know how to solve this. We'll just put it in diethylene glycol. Well, diethylene glycol is great if you're just a chemist and you're trying to get this thing to go into solution. It is bad, though, if you're going to then give it to small children because it will actually kill some of them. So 107 people died due to this, and that's what really spurs the FD&C Act. And then I'd say the other big caveat that kind of comes in there for the way the current clinical trials is set up is in 1966, we had the thalidomide scare from Europe. So Europe had this drug they were using, they wanted to bring into America. And the FDA at the time said, something doesn't sit right with us about this. We're actually going to set up some standards around safe and effective before we let this drug in. And that was huge because this drug ended up causing all sorts of birth defects in Europe. And this to some degree leads to that FDA process that we see today. In general, the FDA is kind of set up like this right now. For the most part, the stuff we're interested in is the portion of the FDA that regulates medical products, i.e. biologics, drugs, and devices. And therapeutics like CRISPR kind of get rolled into the drugs and biologics and get regulated in a very similar way. One big thing to remember in all of this is that the FDA is a government legal entity that's attempting to use science and a lot of statistics in order to do public good by making products that end up in the medical space safe and effective. The beauty of this, and this is something that's changed over time, right? It's not a perfect process, not completely set in stone. It's pretty well hammered in stone, but it's dynamic and can change in response to public need. And we even see that with these therapeutics with CRISPR that are coming out right now. The FDA is very amenable to trying to figure out how to bring these about because they're not like anything before, to make them safe and effective, to basically get the public good out of it with mitigating the risk, which is not an easy thing to do, but that's part of the beauty of having this kind of dynamic system. So if we look back then at this federal clinical trial overview process, we can dig in a little bit deeper. So in the first half of it, you've got to identify what your target is for your drug or your therapy. In this case, we're going to specifically look at sickle cell tonight and how different CRISPR therapies or targeting can go about potentially solving sickle cell. We'll then go from there. You enter into, after getting some paper again with the FDA, you can enter your phase 1 clinical trial, which is basically to show that what you are using is safe in humans, and it's a very small subset of people. You then move to phase 2 if that goes good, grow up the number of people, make sure that it's still safe, and show that it is slightly effective for what you're hoping to solve. And then phase 3 is where the bulk of the focus is. This is expanding the number to get statistical power, show that it is safe, no side effects, no weird side effects, and that it is effective doing what it's supposed to do. Once that's all together and you've had the FDA basically get into your manufacturing, make sure that all the things you are making can be made over and over, that there's quality control set up, CGMP, stuff like that. You can then apply for licensure. Once you get through that, you can then go to the marketplace with your drug or therapy. To dig in a little bit of each one of these little steps in the bottom portion of that, basically the kind of the clinical trial process. So the preclinical stuff is, I would say, from a scientific point of view, it's where all the interesting stuff gets to take place, right? This is where that scientific creativity is coming in based upon years of your institution's research and decades of other people's research culminating into this new way to solve a problem. And we see this with CRISPR being applied to sickle cell. We're literally working off of 100+ years of other people's research of understanding how the disease progresses, et cetera, to design these and how CRISPR is going to go through and potentially deliver a life-changing therapy for these folks. This is also in the preclinical where the business planning really takes place. Can you make a business that actually can survive this process of making it through the FDA and have something on the outside that can then make its way into the market? It's where budgeting takes place. It's where investment takes place. You're going out, finding people that are going to basically put up the money to help you get through all of this and bring something to market that will hopefully change lives. This is also where patents go in. So you're getting your patents put in at this point. And this is kind of illuminating because a lot of us look at patents and go, wow, why are patents so long? Why 20 years? Well, if you look at this FDA process, sometimes it takes you 10 plus 15 years to get through the FDA clinical trials. If you're putting your patents in now and getting approved, you're basically just getting a couple years on the backside of getting market approval in order to kind of recoup from all this R&D and time invested. The end of the preclinical process is basically culminates into what we call the investigational new drug application to the FDA. This is on average a 14,000 page document that the FDA has 1 month to basically make their decision on. And actually, you know what, it actually stretches out to be a lot longer than this a lot of times. And there's some nuances that go in there. For something that's trying to get fast tracked, people that are trying to apply can actually give the FDA additional money to hire more reviewers to then make these kind of systems go a little bit faster. But once you get through the IND, that's when kind of the rubber hits the road and you end up in your phase 1 clinical trial. So phase 1, remember, the goal of this is to make sure that it is safe in humans and that the dose is right in humans, because prior to this, all your preclinical trial stuff has probably been in vitro, in vivo, and then maybe in mouse models or other animal models. And so this is really the first time it's going to go into human, where dosage might be different, right? We've done all we can to kind of hedge our bets and get the right place, but it's to confirm both those things, that it's safe and that you've got the dose right. In usual cases, this means you've got participants like up to 100 that might be involved in the phase 1 clinical trial. For the CRISPR related stuff, it's a much, much smaller number. It's actually usually beneath kind of 10. And that's to some degree because of the duration of the phase 1 of just the way the therapy takes place. Usually in most drugs, therapeutics, it's a many months, but in the case of these CRISPR therapeutics, it's actually going to be on the order of years. Your historical pass rate, how many of these INDs that get into phase 1 actually pass through to phase 2 is only about 70%. So that's actually pretty high, right? A lot of new drugs, new therapies get through this process and end up in phase 2. So phase 2 now, we're expanding the number, because when we expand the number, right, we're probably going to be able to see any side effects, which will let us know even more about safety, because we're going to have a little more statistical power. And now we can start looking to see, is this effective? Is this treating the patient in the way that we are hoping to? So interestingly enough, with a lot of the CRISPR-related clinical trials, phase 1, phase 2 is often coupled. And the reason this is because the treatments are taking so long, and the analysis in order to make sure things are safe are going to take so long that they want to be able to follow people for years instead of just a couple months, because the potential side effects are things we worry about over the course of months to years. So we want to make sure that there is time for those to occur and be able to check off and say, okay, those aren't there. The pass rate here drops drastically. Those that enter phase 2, only 33% make it out the other side. And remember, that's because you have 2 gates now. You're looking for safety and you're looking for effectiveness, and you're at a statistical value now where you can start really assessing both. And so that's why that gate all of a sudden becomes a lot more narrow. When we get to phase 3, and phase 3 for a lot of the CRISPR stuff is probably another two years off in a lot of these clinical trials, especially for sickle cell stuff. It's now to increase the number enough where you have statistical power to really be able to look at the effectiveness and make sure that the safety is still there once you get to a larger number. This also allows you to expand kind of the demographics potentially that are being looked at for the safety and efficiency of this. This is really famous if you look actually at the SARS-CoV-2 vaccines. Like their phase 3s, and I think Moderna's was like 15,000 in the control group and 15,000 in the group that actually got the vaccine. And then they basically did a demographic that split all across with the demographics of the United States for everybody kind of 18 and older. With the clinical trials for sickle cell, this is going to be a much tighter kind of demographic, just because the tighter demographic of who these usually affects. The historical pass rate here tends to be 25%. So if we do the calculation, right, multiply all of these percentages together, what it's like to come through the whole phase 1 through phase 3 clinical 3 trial process. You go 70% times 33% times 25%. You're now at 6% of people that enter phase clinical trials actually exit out the other side. So this is a pretty rigorous process. And through this entire time, the FDA is having dialogue with you about what's going on, especially with these CRISPR ones. It's really, really interesting to see the dialogues back and forth. On the other side of this, people get really focused on the clinical trial portion, because this is really where you're like, okay, there's hope in this working. But at the same time, the FDA is all up in how you are basically doing the production of your drug or your therapy. So they're in there making sure you've got CMG employed, you've got the right QCs employed, you've got different ways of looking at your reagents, making sure your reagents are stabilized coming into the process, and making sure that when you do the scale up, that all of those things stay in place. So that way, if they are eventually going to green light this, they know that the manufacturing isn't going to change and accidentally change that drug or that therapy that comes to market. So once you have all of those ducks in an order, that's when you can actually apply through the new drug application for licensure or approval. And what this approval basically gets you is a rubber stamp that says you can now market this to the public with the proposed label. And the label basically is, here's how you use it, and here is all the risk that we know about it based upon the phase 3 clinical trials. And just FYI, if you throw out that original, the IND was big, the 14,000 pages, this puts that to shame, because this is all of the FDA paperwork about your manufacturing and the clinical trials and the preclinical trial stuff. We're talking truckloads of paperwork, and the FDA has 180 days to make this decision, which this is usually where something's getting fast tracked. This is where the money is coming in to buy more people on the FDA staff to kind of come in and look at something. And also, this is where a lot of negotiation takes place between the FDA and the people, the sponsor basically of the drug or therapy, to basically say, hey, we're gonna give you more time with this if you can give us a little bit of patent extension. So patent extensions are one of the major tools, one of the biggest thing that sponsors are looking for is kind of play points here. And that's because every little bit of time you add on is basically more time you have to recoup all the investment that you put in. And there's a couple of things that the FDA kind of bakes in that you can do during the clinical trial process to basically guarantee yourself some more patent time. One of those is a pediatric data set. You can get a 6-month extension if you have a pediatric data set, and that's usually coming in during phase 3 clinical trials. And we actually see that with some of the sickle cell therapies that are kind of in this pipeline right now. The one coming from UCSF, UCLA, and IGI has that baked in there. Phase 3 has a pediatric set in there. And that's actually really important, because if you really want to help people with these therapies, you want to help people with sickle cell as early as possible, because you don't want them to go through this painful life to only get therapy in their 20s or 30s, when you could be solving this like as soon as it shows up. Also, the other kind of carrot that the FDA can wiggle out there is if you're solving a problem for under 200,000 possible patients, you can negotiate for additional months of patent extension. So sometimes these are known as orphan drugs or orphan therapies. Sickle cell kind of plays on the edge of this a bit. So in order to actually kind of dig into the therapies we're going to talk about, I need to get you guys kind of at least conversational about CRISPR, how it works, so we can understand how it's being deployed in order to try and solve sickle cell disease. So what is CRISPR? CRISPR is a elevator pitch, genome editing tool that is highly precise, cheap, and effective compared to the stuff that we used to have before. So we've had genome editing tools for a while. Actually, most of you guys already teach them. They're called restriction enzymes, but they're not very specific. Sometimes they're kind of clunky to work with. And since restriction enzymes, we've built better ones called zinc finger nucleases and talons that actually could give you specificity to go to really exact spots, cut and edit there, but they were a pain to work with and really expensive to deploy. CRISPR revolutionizes this like tenfold, hundredfold. But what does CRISPR actually mean? CRISPR, if we break it down, means clustered, regularly interspaced, short palindromic repeats. That's a mouthful, and that's why we just use CRISPR. The other thing you also hear associated with CRISPR all the time is this word Cas. And Cas just stands for CRISPR-associated protein. And this is usually a nuclease, usually something that's going to go through and be able to do a cut in DNA. So CRISPR was first discovered in bacteria, and it was realized that what this was, these clustered, regularly interspaced palindromic repeats, well, in between all of them, they had little pieces of phage DNA. And through kind of scientific investigation, people realized what was going on was what we were seeing was an adaptive immune system that bacteria had. So basically, when they get infected by a phage, they could take that nucleic acid, they could break it down, and they could store little bits of that in between these palindromic repeats in their genome. That little piece of phage DNA would then be translated into RNA that can dock with one of these Cas enzymes, those nucleases, and they could then do surveillance of the cytoplasm looking for nucleic acid that looked like it came from that phage. And if they find it, they would go there and cut it. And so biotechnologists saw this and went, wow, that's a pretty cool trick. In fact, if we could gather this technology, we could then really edit genomes really easily, because if we could just program that RNA piece, we could then send a nuclease after just about anything we could possibly imagine. And so that's exactly what's taken place through like Jennifer Doudna's group and Charnay. And so what we now have is this technology where we can basically get one of those Cas proteins, we can give it what we call a guide RNA, basically the hunt and seek function. And this Cas enzyme now can look at nucleic acid, find something that's complementary, and then drop a cut right where we've told it to go. It's kind of like the ultimate control find, copy and paste for genomes. And it's really easy to use compared to all the systems that have gone in the past. One thing to be aware of, though, when we're talking about CRISPR, when you hear about CRISPR, especially kind of like in media and sometimes even in scientific literature, is there's often two things baked in when people say, I'm using CRISPR to do something. There is the CRISPR, the using of basically the Cas protein and some sort of guiding RNA to go cut someplace. But then there's also usually some sort of repair mechanism on the other side of that then is doing the modification for a repair or a knockout sort of scientific investigation. So there's two sort of repair mechanisms that get used frequently with CRISPR that you need to just be aware of. There is what we call homology-directed repair. And this is what your cells do. If you get a double-strand break, your cells basically look around and go, oh, hey, there's the other copy of that gene or that allele. I'm going to take that and use that as a template to then restitch where this double-strand break took place. Well, we can do that. We can actually deliver a piece of DNA that looks like the flanking zone around the place that was cut, and we can trick cells into basically using that to then put that DNA into that cut. This allows us to introduce things into a cut site. So say you wanted to like drop a gene into a genome, this would be the way you would do it through HDR. However, there's another route that's a little, not sloppier, but a little more easy to use with, because you don't have to put extra DNA in there. It's just non-homologous injoining. So you've delivered a cut to a genome, and a genome just really quickly tries to stitch it back up together. And usually what happens is you get indels in that process. You'll get an insertion of a base or two, or a deletion of a base or two, or some of the times it just stitches back up perfectly, and it just looks like the original genome. So those are the two kind of repair mechanisms that are always or usually associated with CRISPR when you hear about it talked around in the media. So if we're going to take this technology and go after a disease, what makes a really good disease to be able to apply CRISPR to? Well, first of all, you want something that's really well characterized, right? You don't want to go fishing around in a genome for something you're still trying to figure out. You would also like something that has single changes in the DNA code can have big effects. Sickle cell is one of those, right? And that could be, you're going through and changing a SNP, or maybe you're going through and changing a promoter region, which is also small pieces of DNA where a little change can have a big impact. And then also, we'd like to work in tissues that are easy to work with. Other considerations, and this to some degree is bioethical considerations, but there's also good scientific reasons to think about this, is modifying somatic cells is predominantly what we should be doing for a really, really long time. And that's just because if you were to use CRISPR on germline cells, you're not only modifying that person, you're modifying all of their descendants. And we don't have scientific knowledge about what that might do, what the consequences of that might be. And so that's why any sort of germline modification is something that's really out of the question for these therapies when we're talking about them. Even though you've seen this kind of reach its way to the news with Dr. He Jiankui in China, who we modified, remember, the 2 young children so that they wouldn't be susceptible to HIV. But for the most part, what we're going to focus in on is the somatic stuff, because that's where all the therapies are going to be in our lifetime. If we look at somatic cells and think about how we modify them, I would say there's even 2 more kind of buckets we need to think about. There is in vivo modifications. So we're going to do the modification right inside the patient. I'm going to inject you with something and change the cells right in you. Ultimately, this is where we want to be with cell and gene therapies. But the space we're going to stay in for a while is probably ex vivo, where we actually harvest cells from the patient. We take them to the laboratory and then do the modifications there. But that's because this is leaning upon all of these techniques that we've been building over the last couple of decades and all of our knowledge of control and things like that. And then taking those cells, once we've modified them, once we've confirmed they've been modified correctly, and then regrafting them into the patient. And within this, there's even kind of 2 groups to think about. There's autologous, which is you're taking that exact patient's cells, modifying them, and then regrafting them. And then there's the potential for allogenic ones, where you're taking somebody's cells, modifying them, and then usually stripping out HLA and other kind of immune system markers so that you now have a cell line that could go into anybody. All of the CRISPR therapies so far that are in the FDA pipelines are all autologous ones at this point, but everybody definitely has their eye on getting into allogenic ones at some point. So let's do a little background on everyone's favorite SNP, or at least from a teaching point of view, sickle cell. This is, I think, ever since people started learning about CRISPR, this was always the disease within everyone's kind of focus, because it's very obvious what you could do in order to make a big difference for these patients and solve the disease state. So as a refresher, there's a single nucleotide polymorphism in hemoglobin's beta unit. This SNP changes basically an A to a T, which now gets you from having a hydrophilic amino acid to a hydrophobic amino acid in the structure of the beta globin. When you do this change, all of a sudden you stop getting these nice tetramers of beta and alpha, and what takes place is those modified beta amino acid sequences now start sticking to each other, and you get these long polymers. These long polymers actually deform the cells, giving them their sickle shape. These sickle-shaped cells now can cause all kinds of problems for the patient. For the most part, we talk about a lot of these as being vaso-occlusive events, i.e., you've got cells that clog up some part of the arterial system and then usually result in extreme amounts of pain, if not worse, right? Because if these were to take place in places like the brain or the heart, you're now talking about heart attacks and strokes. But since this is just a SNP, this becomes a really great target for a gene therapy involving CRISPR. If you were to just kind of show this to your students and how CRISPR works, you could probably ask them, what would you do to solve this disease? In fact, in Bio-Rad's kit, we actually give them the chance to do that. So if you just download our manual, there's a capstone activity where students can basically plan how to use CRISPR to modify that SNP that causes sickle cell disease in patients, and then brings them through a little bit of the background how that would take place. The harvesting of hematopoietic stem cells from the patient, taking those out, modifying those, and then regrafting them. That kind of gets them into the nitty-gritty of how you go about solving this. And not too surprisingly, there are groups using this exact sort of strategy to solve sickle cell. So UCSF, Berkeley's IGI, where Jennifer Doudna is, and UCLA are all together on this clinical trial to do just that. Basically use CRISPR and HDR, homology-directed repair, to replace that SNP so that you have the wild type again. In fact, you can find out exactly where they are by going to clinicaltrials.gov and seeing, okay, they've got 9 participants they're trying to recruit for the phase 1, phase 2 clinical trial. Here's the estimated start dates and things like that. Of course, in between, we had a pandemic, which has slowed down a lot of these clinical trials. In fact, if you want to dig in and find more of them, there are quite a few out there right now when we talk about CRISPR being used for a sickle cell therapy. And the way you can find this is go to clinicaltrials.gov, and then there are conditions you can search for. So I'm going to search for just sickle cell, and then I could put in the term CRISPR, and it will bring up all of these basically companies that have paperwork in with the FDA saying, hey, we want to get to the clinical trial level with you guys. And it tells you at what level they're at. So one of the ones that pops up really immediately here is we've actually got one that's completed. So that's the one we're going to deep dive into. It's also the one Victoria Gray is part of. If you change your search functions to go after different types of gene therapies, you'll find that there's other ways that people are trying to solve sickle cell using CRISPR, lentiviruses, all types of things. But really this Vertex and CRISPR Therapeutics one is the one that's the furthest along. So we're going to kind of do a deep dive into there. Anyone that's teaching at the collegiate level or the community college level, though, you might want to dig into Bluebird Bio that's using a lentivirus approach to this. And they've had all sorts of stumbling blocks. And this really gets to some of the risk that's involved, I think, for all of these therapies. I don't have enough time to dig into that right now, but just know that that is there, and it's really worth going through with your students that might be really finding themselves deeply involved in FDA stuff later on. It's a really interesting piece about risk management. What we're going to focus on from here on out, though, is the drug therapy CTX1 by Vertex and CRISPR Therapeutics. Vertex is from Boston, Massachusetts. CRISPR Therapeutics is actually from Switzerland. Charpentier is their CSO, their chief scientific officer there, but they also have offices in Cambridge. They're working with at least 3 institutions in Tennessee, Sarah Cannon Research, TriStar Centennial, and St. Jude's that have a long history of sickle cell work in order to do their their clinical trials. So how did they identify that this was the thing they wanted to go after? Well, sickle cell affects many people worldwide. And so this is a very, very kind of sought after therapy, right? Because 300,000 people worldwide are born with sickle cell disease every year. And just coincidentally, if the way that they're going to solve this problem potentially will also solve other beta hemoglobin diseases like beta thalassemia. We'll just briefly hit that in a little bit. But their approach is very different to the one that we just kind of mapped out earlier and the one that the UCs are working on. And the reason that they decided to go this other route that's really kind of ingenious is due to things that we've been seeing for almost over 70 years now. In fact, the person I would actually kind of attribute this to is a physician by the name of Janet Watson in 1948. And this is not too long after like sickle cell was finally kind of described as like what was going on there. And she was noticing that patients from early childhood didn't have it, but then they would start to develop sickle cell symptoms as they got older. And she was actually able to correlate this with fetal hemoglobin concentration, basically preventing disease state. So we've known about this for a while, but actually understanding the mechanism of what's going on there is what's taken a couple decades to really dive into. One of the other big keystones along this process was people saw that if you have a mutation that more or less keeps fetal hemoglobin in your bloodstream at all times, you can have the genotype for having sickle cell and never develop disease states. So kind of these things combined really kind of pushed a lot of people to start looking at, well, how do we keep fetal hemoglobin around? Because that could get rid of the disease state. And it would solve it for sickle cell, beta thalassemia, and some of these other beta hemoglobin involved diseases. So this is what Vertex and CRISPR Therapeutics is doing. They're going to use CRISPR not to go after the SNP, but to go after an enhancer region for a transcription factor that is the repressor of fetal hemoglobin. So the truncation of that is basically they found a way to take CRISPR to knock out the repressor for fetal hemoglobin so that it will get to circulate in these patients and resolve them of the disease state. There's great scientific evidence for this working. In fact, we actually kind of saw the tip of this with Victoria Gray's story. At 3 months old is when she had her first sickle cell disease crisis event. And this is something that we've seen as we track people that have sickle cell, is that After birth is when your fetal hemoglobin starts to drop off. And right around 3 months is when fetal hemoglobin really drops off and your beta hemoglobin is supposed to take off. If you're sickle cell, this is the point at where you're getting more sickle cells and getting your first crises events. And so this is what this therapy is going after. BCL11A is really interesting. It's a transcription factor that's associated with the chromatin remodeling protein family, BAF, SWI, SNF family. These are all ATP-dependent chromatin remodeling stuff. Ironically, I worked on this at NIH at the time. Remember, all primary research eventually leads to something, even if you don't see it at the time. But what they found was that some of the enhancers for BCL11A could specifically modify whether or not fetal hemoglobin was produced or not in rat models and in rat models at this point. This was discovered to some degree that this might be a really good target through genome-wide association studies in the early 2000s. So human genome's out, people start doing these genome-wide association studies. And one of the ones that people were really interested in were these people that had basically fetal hemoglobin as adults. This allowed us to target this specific transcription factor. Once that was on everybody's radar, people started going through and really trying to understand how enhancers changed what this transcription factor does. Because remember, it's a transcription factor. It touches potentially a lot of things. And through these studies, one of these really key ones that's super, super cool and actually did use CRISPR to figure it out is they went through and they just designed basically CRISPR for all of those sequences where it could land within the enhancer regions for the transcription factor. And they found that there was one that if CRISPR went in there, cut it, and basically knocked it out, what happened was that all of these cells would start producing fetal hemoglobin and that nothing else was going on. So they basically found how to tweak this transcription factor just so that you could get it to produce fetal hemoglobin. And this is ultimately what gets baked into the therapy that CRISPR Therapeutics and Vertex is using and that Victoria Gray benefits from. So they then designed basically a guide RNA that targets just this, what they call the erythroid enhancer region for the transcription factor. If you can go in and basically just deploy NHEJ, the non-homologous end joining, and get an insert or a deletion in there and get this thing so it doesn't work, all of a sudden you have fetal hemoglobin circulating. And this is what makes the life difference for Victoria. Let's back out of all the scientific stuff for a bit and think about what it's like to be the patient. So I think this is going to be really important when we talk about not only this therapy, but a lot of therapies that are going to be using CRISPR, especially in an ex vivo format. So Victoria has to go to Tennessee months before she's going to get therapy. And that's because during that time, she's going through apheresis. She's getting a drug to stimulate basically hematopoietic stem cell production into her bloodstream, and they are drawing tons of blood from her, partly so they can grab these hematopoietic stem cells, but also so they can bank her red blood cells, because those are going to be critical for after she gets her graft. She'll go through myoablation. So they use busulfan to do this with. That's basically chemotherapy, and it destroys all of the bone marrow. And there's a bunch of caveats with this, because this is one of the things that gets used for a lot of these different therapies that are ex vivo. I think it's one of the things we'll probably get a lot of scrutiny in the future. So she goes through myoablation. She gets now the infusion with her own modified stem cells, and then those now have to graft. So they're floating around her bloodstream, and luckily for us, those cells know exactly where to go. They'll go back to those places in the bone, they'll regraft there, and then she's basically on a transfusion of her banked blood cells for about a month. So this is a pretty intense process. You're very, really brave to want to put yourself through this. And it's extremely a lot of effort on everyone's part, the patient, the medical staff, everybody involved. If we now look at the medical, or not the medical side of it, but the manufacturing side of it, we've got this going on. The apheretic material is coming in. We're isolating just the hematopoietic stem cells out of it. The manufacturing of the Cas9 with the guide RNA targeting just that enhancer region is being electroporated into those cells. Those are all being stored. Then they do flow cytometry and sequencing to make sure that the modifications they're getting are what they want. There's no off-target stuff going, nothing weird going on. and then basically confirming that before they can build the infusion for Victoria. And here we are, July 2nd, 2019, Victoria's on her infusion day. Everything goes smooth. Everything goes really smooth. In fact, we're at least 2 years out now. She's had no hospitalizations. She's off the pain medications. I mean, life-changing. She said, I don't have to worry that I'm going to end up in the emergency room when the weather changes. I'm not tired and drowsy from taking all these narcotics. I can make plans, just regular plans to go on vacation and do things with my kids. So for the first time in her life, she actually has the potential to have a normal life. Prior to this, prior to going in for the transfusion, she was ending up in the hospital at least 7 times a year for multiple days to do pain management. It would be sporadic. Also suffering from what we call pain insomnia, where you basically, due to pain, you're just waking up all the time. So on top of having all the pain, you're also just sleep deprived your entire life. So for once, there's some light at the end of the tunnel that she can live a normal life. She's been followed up with monthly after this. Blood draws are being done to kind of see what's going on at the kind of scientific level. And what we're seeing is that if you go through and you look at her blood fractionation for fetal hemoglobin, it's there and it's steady. And it's been steady now for about 2 years at least over 45%. Same thing if you go and you look at the circulating cells and ask how many of them at least have fetal hemoglobin being produced at some level, over 99% of them. This is really, really effective that this got in there. But really the proof in the pudding is this. Since that infusion, she has not been back in the hospital for any sort of treatment due to pain or any other sort of vaso-occlusion, those capillary clogging type of events. So game changer. In fact, her story now has definitely recruited a lot more people to come in and want to be a part of this clinical trial. If you really want to get into the story of this with your students, and this is really great, especially for I think high schools, to kind of get there and conceptualize this. NPR did a number of shorts on her throughout this process. And actually the easiest place to find those is actually on the IGI, the Innovative Genomics Institute website, but they kind of step through every single process in there. So it's a really great way to get the emotional human connection of why these therapies are groundbreaking. And then if you're looking for even more, you want to dig in even deeper because we just don't have the time to dig super deep here. There's a number of other ones out there. There's a really great interview with somebody who heard Victoria Gray's story and then basically jumped in and got himself into the clinical trial. He's very articulate in the way that he talks about the pain before, what the process was like. You can find that on CRISPR Cuts. And then for the colleges and the community colleges, there's 2 YouTubes I would kind of recommend. There's one that is UCSF and UCLA and IGA talking about their process. And what's really cool here is it digs into the dialogue between them and the FDA. This is something you were never probably going to hear out of any of these other companies because they're private entities. They're keeping that as part of their protection, where this is very out in the open. So you really get to see what that dialogue is between the FDA and the sponsors of the therapies, which is super cool. And then if you want the deep, deep dig into the BCLA story, Professor Stuart Orkin does a fantastic talk of all the science that builds up to these therapies being possible. Those last 2 were also really interesting because they talk about why this is not the end all be all, what the risks still are, and kind of what the perspective is beyond these. But I don't have time to dig into that, but just know that those are there if you want to dig even deeper. So those are really important for showing your students why these things are transformative. But in order to really take them through that, I think they actually need to have some sort of experience where they can experience CRISPR. And so Bio-Rad has an ability for you to do that now in your classroom in their out of the blue CRISPR kit. And they're doing a very similar thing to what's going on in sickle cell. We're going to use CRISPR to do some sort of knockout that's going to change basically a phenotype. We're not doing fetal hemoglobin in your lab. That's a little bit hard for everybody to diagnose, but what we're going to work with is something that's colorimetric. Basically, we're going to take LAC-Z, the ability to metabolize lactose. We're going to knock that out and use a reporter mechanism that's known as blue-white screening to show that you've done that. We'll use, instead of just NHEJ, which has been used in the Vertex in CRISPR Therapeutics, we're going to use HDR. So that way they get to see the full tool set when they do this lab. And what they're going to do is they're going to use CRISPR-Cas9 with a guide RNA that's going to be directed just at that LAC-Z gene. What they're going to do is they're going to be able to deliver a double-strand cut, and then using HDR and the ability to basically turn on and off a repair mechanism, they'll be able to show that they can use HDR now to stitch a stop codon right into the middle of LAC-Z, which basically knocks out its function. And so what you will have then, you'll have a blue colony that all of a sudden isn't able to metabolize lactose and produce blue due to the presence of XGAL, and it will be a white colony at the end of this. But since we're just doing a blue-white kind of selection here, and for the most part, the technique to get the DNA in there is very similar to other things students might be involved with, which is like bacterial transformations. The controls of this lab are really the focus point, I feel, that allows students to understand that what they are doing is CRISPR and actually understand the nuance of what goes on in CRISPR. So in order to understand the controls, I need to give you a little bit of background on those. So we're going to be using E. coli that a double-stranded break causes lethality. Basically, you go in there with CRISPR-Cas9, you do a double-stranded break, that thing is dead because it can't repair itself. The other thing we're going to be using is we're going to have Cas9 just expressed all of the time in these cells. So that way you don't have to worry about delivering Cas9 into the cells. And then the other thing that we're going to be able to have control over is the ability to turn on or off the repair machinery. And this is basically going to be able to make the bacteria survive or not survive the ability of one of those double-strand cuts caused by CRISPR. And so what this looks like in your students' hands is this. They're going to have two different plasmids that they're going to use bacterial transformation to get the DNA inside of the bacteria. And then through those controls, they'll be able to figure out how that has either caused CRISPR-Cas9 repair to take place or not. So the first basically plasmid just has donor DNA on there has donor and guide RNA on there. So this has the guide RNA to show where to cut, and it has the donor DNA in order to do the repair to drop a stop codon in there. The other plasmid they'll work with just has donor DNA. So it doesn't have the guide, so the Cas9 can't functionalize its CRISPR and go and cut in place. Ultimately, what students do is this. They're going to take bacteria that start off with their repair machinery turned off, or they'll start with bacteria that have their repair machinery turned on. And they're going to basically move both of those plasmids into these two different cell lines. So if we look at just the ones that have the repair system turned off, if we were just to give it donor DNA, so just the DNA, if there was a cut it could repair it with, basically nothing happens because there was no cut since Cas9 had no place to go and cut. And so what they'll get is they'll get blue cells at the end of this. If they move in that other plasmid, right, the one that has the guide RNA that can show Cas9 to go cut in the LAC-Z gene, and it has the donor DNA, so you could have the DNA to stitch potentially. But you got to remember, this cell line doesn't have the repair machinery turned on. So what happens is Cas9 grabs the guide RNA, it cuts in LAC-Z, but since that repair machinery isn't there, you have a double-strand break and it causes lethality. And so you won't get any bacteria on this plate because they haven't survived being cut by CRISPR-Cas9. We then move on to the other lineage. We then have cells that have their repair machinery turned on. So if we give them the donor DNA, they've got the donor DNA, but they can't do a cut, cells just end up the same when you replate them, they're blue. This last one though, is where you've got all of the pieces in place. You've got a plasmid that has the guide, so it can hybridize with the Cas9, it can go cut the LAC-Z. You've got the donor DNA and you've got the repair machinery turned on. So what happens is you're able to deliver a double-strand break in LAC-Z. You're able to do HDR and put in a stop codon, and it's going to be able to repair those bacteria survive, but now they have a non-functional LAC-Z, and we're able to see this just because they are white at this point. And so that's just kind of the high-level look at what that looks like. There's different ways to talk about that with your students, but ultimately this is what they're looking at the end and looking through their controls to then demonstrate that what they've done is actually CRISPR. And the beauty about this is you literally just need a water bath, the ability to make hot water, and an incubator in order to pull this off in your classroom. You don't need any fancy tools, and this helps them see the power of CRISPR as compared to a lot of these other types of genome editing tools that have been used in the past. If you're working with kind of a higher level group of students, they might go, well, I'm not still convinced that this is CRISPR. And so you've got one more tool set that you can pull out and play with them. And this is Bio-Rad offers a genotyping extension kit so that you can actually genotype your modified cells to then confirm, help them confirm that what has taken place has been a CRISPR modification. And so this is by far the easiest PCR you'll ever do in your life, because it's just going to be colony PCR. You're literally going to scoop up cells, put them in your PCR mix, go on your thermocycler, run through the process, and then throw it on the gel, which makes this really easy to pull off. The way that we've engineered this so that you can prove that what's going on is the delivery of that stop codon is you're going to be doing a multiplex with 3 different primer sets. And so you're going to have primers that are going to look at 3 locations. The first one's going to look at basically unmodified LAC-Z. So you've got a primer that's going to land actually where the cut site's going to be taking place, and then a primer that lands further down in LAC-Z gene. This will give you about 1,100 base pair amplicon. Your second primer set that's also going to be in this mix is one that lands inside the insert. So if you inserted the stop codon, it's going to land in there, and then it's going to amplify out to give you a 650 base pair amplicon. And then the third one is, of course, like any good scientific process, a control. And so this is going to be a primer that lands outside of all of that stuff that just shows you, yep, I got my DNA there, something else isn't going on, PCR worked, check, check. And what you can then do is, after you've amplified those, is run those out on a gel with your students. And what you can look at is cells from, or colonies from, those different plates. So from plate C and then plate D and other ones if you are interested in them. And then you can compare those bands to then demonstrate, hey, what we've done is actually dropped a stop codon, or dropped something in there that's preventing LAC-Z from working by doing the band analysis. And so this kind of gives you the whole toolbox really to do CRISPR and do CRISPR genotyping in your classroom, which basically sets them up for really understanding how these therapies come to market, what that base technology is like. And then there's extensions within the kit. So after they've done these processes, there's these kind of capstone projects. We already talked about one of them, which was actually looking at the SNP for sickle cell and how they might design a guide RNA to go about and target that. But then there's also 2 other capstones that have them look at how could you develop a CRISPR therapy for cystic fibrosis, another one, right, that is a very, very well-documented SNP, and then another one for coronary artery disease that's a little more complicated and has a little more nuance to that. But this really gives them the whole picture of what it is to work with CRISPR, to do these modifications, to then potentially bring more of these life-changing cell and gene therapies based in CRISPR to the marketplace. If you need any of those tools, you can get them at biorad.com/teachingCRISPR there. There's also a great hands-on kind of manipulative model that shows them the mechanism of how CRISPR works. And you can download that for free. You don't need to buy a kit or anything. And you can use that with your students. So that way they can at least get used to the mechanism, and also helps them do the statistical modeling about why CRISPR is so powerful, in addition to its ease of use. And with that, we're almost out of time. So I'm going to go ahead and stop and see if there are any major questions that I should tackle out of the Q&A. Otherwise, with a lot of those Q&As that came through that weren't able to be handled in the chat today, what we will do is we'll send out a follow-up email that will go after some of those and give you a little more background on them. So with that, let me see if there's...
Damon?
Yeah.
We left some questions that
okay.
are in the Q&A, and that would be awesome if you could scan those and
yep.
answer those.
Yep, you got it. I brought those up, and I'm going to basically walk through them one by one. Looks like we've got like 10 or so open of those. And just FYI for anybody that's on a time crunch right now, we are hitting the half-an-hour point. I'm going to continue going over just to be able to answer these, but if anybody's got to leave, know that you're at that time right now. So the first one here is by Emily Neal. How does somatic cell changes alter or treat STD patients to impact all blood cells produced? Which somatic cells are being changed? So the somatic cells that are going after here are hematopoietic stem cells. And so these are the cells that are in your bone marrow that are normally giving rise to red blood cells. And so if you clear out a patient's entire bone marrow with the cells that are normally producing that, and then regraft the modified cells in there, all the cells that are in the bone marrow now, when they produce red blood cells, are producing ones now with fetal hemoglobin. However, though, a big question is like, well, how do you get to 100%? Barely anything gets to 100%. Well, the beauty is with basically sickle cell, you don't need to necessarily have 100%, all of that sickle cell shaped beta globin is gone. You just have to kind of get past a tipping point. And that tipping point is really, at least according to the UCSF, UCLA talks, is probably only somewhere around 30%, if that. So you really only need to kind of tip the boat that far in order to rescue the disease state. All right. Melissa Davis, do they use new Pagan to stimulate the stem cell production? No, they're using, I think it's Plexiflor is what they're using in at least the Vertex and CRISPR Therapeutics one that we talked about in detail here. Beth Rojas, assuming these results are better than hydroxyurea. Yes, much better than hydroxyurea. So anybody that's not familiar with sickle cell, hydroxyurea is kind of the go-to drug regimen that's been getting used as of late. And interestingly enough, we don't really fully understand the mechanism of hydroxyurea, except what it seems to do is it stimulates fetal hemoglobin production. And so what it goes after and what this CRISPR therapy by Vertex and CRISPR Therapeutics is basically going after the same target. How do we get more circulating fetal hemoglobin? But the beauty, of course, about the CRISPR, though, is you don't have to be taking that drug the rest of your life because you are now the one producing it. You don't have to continually stimulate it. The caveat there, though, is that hydroxyurea, even though it's not as good at getting rid of the crisis events as at least what we're seeing with Victoria Gray is, and some of the other patients in these clinical trials, is it's easier to deliver, right, to hundreds, if not hundreds of thousands of people really quickly because it's a drug. Where Victoria Gray, we're talking a multi-month process just to get the treatment. So imagine trying to scale that up for hundreds of thousands of people. And that's why drugs are something people are very interested in for solving sickle cell, or ultimately some sort of in vivo sort of therapy, because the labor involved in getting this to all the people that need it is extreme. And then also the cost associated with that ends up being probably about a little over $1 million per patient if this gets to market. Laura asks, is Victoria Gray still producing mutant beta globin as well as fetal? Yes, she still has some degree of the beta hemoglobin with the SNP in there for sickle cell, but it's being overwhelmed by the fetal hemoglobin enough that she doesn't have any of the disease state. Just in the same way that kind of if you're heterozygous, right, for sickle cell, you will have a little bit of it around, but it's not getting you into the disease state. Scott asks, I was wondering, what is the ratio of donor DNA to guide RNA in this experiment? Is this ratio important for success of the work? I'm not sure if you are referring to Bio-Rad's kit or the Vertex one. In all of these, yes, the concentration, the ratio of donor DNA and guide RNA are going to be really important. None of those numbers for ours or Vertex are easily accessible to me right now. So sorry, I don't have a strong answer for you there. What about CRISPR for antibiotics? Yes, there is a number of different CRISPR therapies that are being used to go after antibiotic resistance. I can't really dig into that talk right now. I did do that actually in last year's talk for NABT. So if you send me a follow-up email, I can dig that out for you. There are some really novel ones of those going on, or you can find them yourself by going to clinicaltrials.gov, typing in antibiotic resistance and CRISPR, and you'll be able to find the ones that are in the FDA clinical trial process at this time. Another question here, HIV patients can be cured with this, question mark. Potentially, there are definitely ways that a similar pipeline, right, could be developed for HIV and for a number of different disease states that could be solved by basically harvesting stem cells, manipulating them in order to make cells that when they are reimplanted could avoid disease states. So the pathway that you're seeing being laid out here for sickle cell will be a pathway that will be walked many upon many times by other therapies going after other disease states. Could CRISPR be used for viruses to integrate themselves into one's genome, like HIV? Mary McDonald, yes. And that kind of goes to that last question a little bit. Will these questions show up in the recording? Yes, it'll be sent later. Yes, these will get in there. Would it be, Austin asks, would it be better to edit out the mutant code that makes the incorrect amino acid with the sickle cell than inhibiting BCL2A? That's a really interesting question. And that's one of the things I think we will learn over the course of the next 5 to 10 years. And that's mainly because there are multiple groups going after using CRISPR, right, to solve sickle cell. I just kind of gave you the tip of the iceberg on 2 of them, the UC version of it, going after editing the SNP, and CRISPR Therapeutics, Vertex, going after this enhancer. There's other people that are going after BCL2A in a different way, which is going after where it lands in order to, have its effect on fetal hemoglobin. So there's a handful of different targets. My one worry about going after the transcription factor is that it's a transcription factor, right? And so we may see different sorts of events take place when this moves into a pediatric group. And that's why I think the SNP modification that the UCs are doing might have less risk. But really, this is really hard to say at this point, based upon all the scientific evidence we have. Both of them seem to have the exact same risk profiles. This will be something to really watch over the next 5 to 10 years. One thing is, though, at least we've got patients at least 2 years out now, like Victoria Gray and a few others, from these original therapies. And so if some of these things are starting, some of these side effects might start to show up, they will be here very, very soon. Is there information on how treating STD patients will affect the heterozygotic advantage against malaria? That's a really interesting question. I didn't run across any data for that right now, but a lot of that we could already probably gain by looking at people that had those original fetal hemoglobin mutations where they had fetal hemoglobin circulating as adults. There's been studies on those people at least since the 70s. And so somebody in those studies may have covered that, which then tells us how these treatments might actually affect the heterozygotic advantage against malaria. Those are all the ones that I think I can cover with the time that we have. There's a bunch of other ones that came on in there that are more kind of questions about getting things about this seminar and things like that. If there are any other ones that really stood out, Aaron, that you want to bring to the forefront, I'll handle those right now. Otherwise, I want to say thank you for everybody for joining.
I just want to say these questions were really good questions that made me feel like I need to go out and do more research. So thank you for those that had some questions. Are we able to maybe answer these offline?
Yep. So we will have we'll have a dialogue of all of these ones that I can capture. And what we'll do is in the follow-up email, I'll go through and try to address these ones that came up, the ones that have actually been answered in here and the ones that are still open or dismissed. We can kind of go through and try and highlight some of those. So and this is great because these type of technologies, right, what they really should do, and a lot of us, is generate just a lot more questions about how are they working, how do we mitigate risk, how do we understand risk, things like that. And so we want to just kind of be able to kind of continue that dialogue with you. I think the easiest way to do that is sending up a follow-up email that goes after all of these with some more time for kind of thought and consideration, point outs to other helpful documents that you can get deeper into some of these questions with. So with that, I want to say thank you to everybody that spent an hour plus with us today going through this. Hopefully you got a lot out of this. And remember, at Bio-Rad, we're here to kind of support you bringing these technologies into your classroom so that students can have hands-on access to understanding them. And we're here as collaborators with you in your classroom. So if you ever have any other questions, you can reach out directly to anybody on the Bio-Rad team. The bioradexplorer.com email will get you to everybody, or you can email people individually. Thanks once again for spending the evening with us, and have a great night. Take care. Bye.